Today, we're going to tackle a really interesting question. How can the food you eat actually change the way your genes behave? We're going to be looking at the nutritional modulation of the epigenome. So here's how we'll break it down. We'll start with the essential puzzle of gene expression. Then we'll get into the key epigenetic switches that control our genes and see how nutrition directly fuels them. We'll see this in action with a classic case study, clear up some common pitfalls, and then wrap up with a quick review. Okay, so let's frame the problem. Have you ever wondered why two people with the exact same genetic risk for a disease can have totally different health outcomes? You see this all the time in fields like nutrigenetics or oncology. Well, the answer isn't always in the genes themselves. It's often in how those genes are expressed. And that answer, it lies in a set of mechanisms that act as a bridge between our environment, including what we eat, and our actual genes. These are what we call the core epigenetic mechanisms. This whole field of study is called epigenetics. Now, the most important thing to get here is that these phenomena control which genes get turned on or off without changing the actual DNA code. Think of it like someone writing notes in the margins of a book. The text of the book doesn't change, but how you read it does. All right, the first and probably most studied of these mechanisms is DNA methylation. This is literally a direct chemical change where a tiny molecule, a methyl group, gets stuck onto a specific spot on the DNA. When that happens in the gene's promoter region, kind of like its on-ramp, it usually acts as an off switch. It just silences the gene. Our DNA isn't just floating around. It's coiled up really tightly around these proteins called histones. And the second epigenetic mechanism, histone modification, is all about making chemical changes to those proteins. This can cause the DNA to pack itself together super tightly, which silences the genes inside. That's what we call heterochromatin. Or it can cause the DNA to loosen up, which makes the genes accessible and active, and that's called euchromatin. And finally, we've got non-coding RNAs. For a long, long time, scientists thought this stuff was just junk DNA, but it turns out they're actually critical regulators. See, after a gene gets transcribed in the messenger RNA, these little non-coding RNAs can basically intercept it and stop it from ever becoming a protein. It's a whole other layer of control that happens after a gene has been switched on. So this all begs a pretty big question. Where do these chemical tags, like the methyl groups we just talked about, even come from? Well, the answer brings us right back to our diet through what's known as the nutritional epigenetic pathway. The perfect way to understand this is to look at the methylation cycle. It all starts with nutrients in our food, things like folate, B12, and choline. Our body uses them to make a really important molecule called S-adenosylmethionine, or SAM for short. Now, SAM is the universal methyl donor. It's the molecule that actually carries and delivers the methyl group for DNA methylation. After SAM donates its methyl group, it turns into another molecule called SAH. And this is key. The ratio of SAM to SAH is what determines a cell's overall ability to methylate its DNA. So which foods actually fuel this cycle? Well, the major players are things like folate, which you get from leafy greens, vitamin B12, choline found in eggs, betaine, and also the mineral zinc. These nutrients are really the essential raw materials for our entire epigenetic system. And if you want to see just how much this actually matters in the real world, just look at choline. An incredible 90% of the US population doesn't get enough of it. And this isn't some theoretical problem. It's directly linked to real health issues like fatty liver disease, and importantly for our discussion, altered DNA methylation patterns across the entire genome. Okay, so let's look at a classic study that really demonstrates this link between diet and the epigenome. It involves a specific type of mouse called the agouti mouse. So these mice have a gene called agouti. When this gene is unmethylated, when it's active, the mice have a yellow coat and are genetically set up to become obese and develop diabetes and cancer. But here's the amazing part. When researchers fed pregnant mother mice a diet rich in methyl donors, those same nutrients we just talked about, their babies were completely different. That diet gave the mothers the building blocks to methylate and silence the agouti gene in their offspring. And the result? The babies had brown coats, they were lean, and they were perfectly healthy. The implications of this study were, well, they were huge. It was clear proof that a mother's diet can directly program her offspring's epigenome. It also showed that a simple nutritional intervention could turn off a gene linked to serious disease. And most importantly, this effect was passed down to the next generation through these epigenetic marks, not through any change to the DNA itself. Now, as epigenetic testing becomes more common, it's really important we clear up a common misunderstanding. And that's the confusion between your chronological age and something called your epigenetic age. So, what is epigenetic age? It's a biomarker. It's a number that's calculated by looking at the methylation patterns at hundreds of specific spots across your genome. And while it does tend to track with your chronological age, how many birthdays you've had, it is definitely not the same thing. What's actually more useful is a metric called epigenetic age acceleration. It's pretty simple. It's just the difference between your epigenetic age and your chronological age. If your epigenetic age is higher than your actual age, that's called positive age acceleration. And we know it's associated with a higher risk for disease and mortality. It's a dynamic measure of your biological aging process, not just the number of years you've been alive. All right, let's take a moment to recap the core ideas we've gone over. So let's review. First, epigenetics gives us a critical layer of control over our genes without changing the DNA sequence. Second, our diet, especially foods with methyl donors, provides the raw materials for this control through that key molecule, SAM. Third, the agouti mouse study showed us just how powerful these nutritional inputs can be, creating lasting, heritable changes. And finally, we can measure these patterns to calculate epigenetic age acceleration, which gives us a really powerful biomarker for biological aging. Okay, to finish up, let's run through a few questions to make sure these concepts stick. First one, what is the primary thing that determines a cell's ability to methylate its DNA? Question two, in that agouti mouse study, what was the specific molecular effect that the methyl donor diet had on the agouti gene? And for our last one, let's think about a real-world application. How might compounds called polyphenols, which you find in things like green tea, influence the epigenome when it comes to cancer? What do you think the answers are? Leave your answers in the comments. And if you like this video, hit that like button. And please consider subscribing to my channel.